talk was also about biomarkers, and I will talk on a similar topic basically, but I will give some statistical background about the case study and the biomarkers, and then I'll go into the computational details. I'm working as a scientist, scientific computing in Janssen Pharmaceutica, and together with as a research fellow in UHasel and KU Leuven in Belgium. So to introduce, let's say, talk about clinical trials and drug development, and normally it's a very slow process because of selecting the endpoints. For example, like if the endpoint is a disease and it's very difficult to measure, then it takes some time to measure it, and then it makes the process difficult. That's why selecting the endpoints is a very important thing in clinical trial and drug development. Now, when we talk about surrogacy, there are two main things. That's the surrogate endpoint and the true endpoint. The true endpoint is the exact response, and the surrogate endpoint is something that you can use to replace the true endpoint. And there has been a lot of research to validate these surrogate endpoints in the past few years. But those research mostly focused on the modeling point of view rather than computational point of view. So I'll talk about that. Now, to a brief description about the biomarkers. So because they are important in clinical trials, and biomarker is something that that's an indicator of a normal biological response to a therapeutic intervention, and that's why it's a surrogate. All surrogate markers are biomarkers, but the reverse is not true. Now, in case of drug discovery, because I'm working in a pharmaceutical company and they are more into drug development. So when they're developing a new drug, they're looking for new compounds and how the compound works essentially. And to do that, you need to also integrate the data from different sources. That brings the high dimensionality of the data and the surrogacy in the context of high dimensional data. Now, as I mentioned that there are different sources, so let's say there is X, Y, and Z. Now X and Y will be the endpoints. So X is the surrogate endpoint, Y is the true endpoint, and Z is, let's say, some treatment, or it can be chemical structure of a compound. So what we are interested in is to check the association between X and Y, but also to take care of the effect of Z. Now, this data is from a project of Janssen, and there it's like the main idea was to check if we can use the gene expression data to understand the mechanism of action of a new compound. So, like I said here, we had three data sources, and it's exactly similar here. So we had the data on gene expression, bioactivity, and the chemical structure of the new compound. The bioactivity data is like a PIC50 or IC50 values of the drugs. Okay. Now, we essentially wanted to model the association between these two variables. Now here XJ denotes the genes because we had the data on gene expression, and we wanted to look if some particular genes are associated with the bioactivity. And like I mentioned before, that there has been a lot of research done in the past, but it was mostly from the modeling background. And as the first speaker also talked about microarray data, so after initially the surrogacy setting started with the clinical trials, but then we moved into microarray data. Okay. About the model, it's a very simple setting. It's just a bivariate normal model, and you want to do inference to look at which genes are associated with the PIC50 values. And we fit the model gene by gene, so that's why there is a multiplicity adjustment requirement. Now going into the computational details. So as I mentioned that it's a gene by gene analysis, that's why if there are more number of genes, then you fit like those many models. So if in this project we essentially had 3,595 genes and 35 compounds, and we had 138 fingerprint features. But this is also a small example. Depending on the experiment, it can be thousands of genes and more hundreds of compounds as well. So with that, the computational time will increase as well. Now for one fingerprint feature and for all the genes, it takes around 6 minutes. Now if you want to do the analysis for all the genes and all the fingerprint features, it takes around 14.5 hours. Now for a bigger project, it will take more time, and that's why we wanted to go into a better implementation of this model. But so initially the analysis was done for all the genes for one particular fingerprint, and now as you can see here that you can compare between genes for that particular fingerprint, but it's not possible to compare between different fingerprints with respect to a particular gene. Now as I mentioned before that it takes almost 14.5 hours to do the entire analysis. That's why we wanted to look at a better implementation, but then there was another motivation to keep the core of R same. Like you all know that if you want to let's say use a low level language than R, C++ or Python, then you can maybe make the process faster, but then you are losing already all the resources available in R, and you need to start coding from the scratch. So we wanted to go for a framework and compare that framework with the existing R packages so that you can already use the core of R and build on top of it. Now to know how to speed up, we actually need to know the code structure and where we are actually losing the time. There are mainly three. We divided all the functions into three categories. One is the GLS, that was basically fitting the model. Then the ANOVA and the summary, these two functions were collecting the results, and there are some other functions which were summarizing all the results in a nicer format. Now if we look here, you can see that the GLS is taking the maximum amount of time of the total computation time. It takes more than 80% of the total time. Now you can again go to the source code of GLS, put some criteria on the convergence, and then maybe speed up a bit, but then yeah, again you are losing the available resources. So that's what was the main intention of our analysis. So when we look into the parallelization, there are mostly two kinds, implicit and explicit. But for the R packages, for example for each, it's implicit because the user doesn't have any control over the parallelization. So you can parallelize the iterations, but it's not like you know which iteration is going where. So that's why the user needs more control over how to parallelize it. Then for explicit, there is some control, but it's not all. And that's why we focus more on the distributed computing in a cluster, and we used a cluster in Leuven, Belgium. Now to parallelize also, there are a few issues that you need to keep in mind, but there are two main issues: are the communication overhead and the load balancing. Now what I mean by communication overhead is when you parallelize, the different processes need to talk to each other, and they can come into like each other's path. So that can like make you lose some time. And also load balancing is the optimized usage of the resources you have. So it's not like you have, let's say, 20 cores and you are using only 10 of them and the rest 10 are sitting idle for some time. So then you are not dividing the entire job in a proper manner. We use the different R packages and also another worker framework that I will talk about. But to compare the performance of all these methods, we use efficiency. And efficiency is essentially the speed gain per core. So it's not like by increasing the core you can always gain a significant amount of time. Okay, so about the R packages, I use for each, cluster apply and cluster apply LB. So cluster apply LB is just the cluster apply with load balancing, and then the worker framework. Now these are the results for the R packages. So with for, of course you see that for all the genes that's the highest, that's the simplest method. But then when we used for each, we noticed that it actually increased a bit, and the increase in time was here for when we did for 2 cores in my laptop, it was less than when we had 20 cores in a cluster. So a possible explanation of this is basically if the way for each is dividing and combining the task, it essentially is making it worse than the for loop. But as expected for cluster apply and cluster apply LB, we had quite a lot of gain in time. But again, it's not really efficient when we move to a cluster and when we are using 20 cores. So you want to have more efficient coding and want to have more speed up. So about the worker framework. Now the worker framework is also a master-slave framework, and so for example, if you have 20 cores, one core works as a master, and then it divides the work into among the 19 cores, and the rest again they finish their job, return back to the master, and then again you summarize the results. But it also depends how you are dividing the iterations. So this is one example where I used 20 cores, but I divided the 3,500 genes in a different combination. So for example, you can have 10 genes to one core, you can have 100 genes to one core, like that. And it's interesting that even if you are using 20 cores all the time, you have different amount of speed up. It's because of the number of job items we have. And of course, the most efficient result is when we have 19 job items, because then we are using all the 19 cores for almost a similar amount of time. And that's why we have the highest efficiency here as compared to the other combinations. This is on the left-hand side, you can see the computation time, and here it shows the efficiency for other combinations. Now, yeah, the levels are a bit small, but here you can see that it's until 40. Now, in the beginning, we had a huge drop in computation time, but later stages, you can see that it's not really a significant gain. So it's not like I was saying in the beginning, that it's not the case that by increasing the number of cores, you'll always have a significant gain. And also you can see that the efficiency is, it varies. And the highest efficiency here is somewhere around when you use 9 cores. So it really depends on you, what you want. If you want a code which takes less time to finish the computation, or if you want like some implementation with the threshold on the efficiency. So that really depends on the user. The different dots here are because we submitted the job few times, and there are small variations when you submit the job to the cluster because of the total workload on the cluster, like how much time, how many jobs are running and how much memory is being used. But yeah, it's not that much. Now, as I discussed, that we did the analysis of the upscale problem, but now there are different ways to also parallelize the job. So for example, we had 138 fingerprints. Now we can select one master and 138 cores to do the analysis for one fingerprint in one core, or we go back to like one of the settings we had with a good load balancing and good efficiency, and we use more number of cores. So for example, in the first case, you see that it takes around 260 seconds, and if we use, but that's with 139 cores, and here with 880 cores, it takes around 97.64 seconds. But when you look at the breakup of those 97 seconds, you can see that it's like 1/3 of the time goes into combining the results. So that's kind of the trade-off you make when you are using more cores. And here we essentially end up using 6 times more cores, but we get only 2.6 times speed up. So it really depends on you what kind of implementation you want to go for. But the good part of it is instead of 14.5 hours, 14.5 hours, we managed to do it in seconds. So that's why we could do some further analysis, as I mentioned before. This was one of the genes that we were interested in, and now we could show that it's associated with all the it's negatively associated with all the fingerprints. So this kind of analysis was not possible in the beginning. Also, we wanted to look for the fingerprint effect on the genes, and then for all the fingerprint features, and that was possible as well. So you can find fingerprint which had positive fingerprint effect and other fingerprint which had negative effects. So earlier, to summarize, as I mentioned, that earlier the analysis was only possible for all the genes and one fingerprint, but so we could compare the genes with respect to that particular fingerprint. But now, because of the parallelization, we could reduce the time and we could do some further analysis. Yeah, it was possible to compare the different fingerprints with respect to a particular gene. We are also trying to do something similar to the worker framework in the cluster, in the cloud. So we are trying to implement the worker framework in Amazon or Microsoft Cloud and compare the performance with respect to efficiency. But that's an ongoing work. And the work with the worker framework is already accepted in Journal of Biopharmaceutical Statistics, but yeah, it's not yet published yet, so I didn't put the link. Thank you for your attention.
Thank you. Do we have a question? A little question? No? You all want to go to the welcome reception. Okay. So, yeah.
You know the futures package? Yes, I know.
Can you repeat the question?
So the question is if I know about the futures package, right? No, yeah. So that's that future package actually it released few months ago, I think, and I'm actually now looking into it. And there is, yeah. So we plan to do something similar with it as well, because this was done already before the future package released. Yeah.
Okay. Another question there.
Yeah, as well, similar to futures that offer the new future, which is much easier interface than Snell and Parallel. There is the RHPC.
Okay.
Try that as well in computation, which supposedly, according to what they declare, it's much faster than cluster LB.
Okay. I didn't know about that, but thank you for the information. It's RHPC, right? Or okay, perfect. Yes, I didn't know about that, but yeah, I was looking into future package, and because we are doing something with the cloud, I was looking into this do Azure Parallel package. I don't know if you know about that, but yeah, I didn't know about RHPC. Thanks. Yeah.
Okay.